No. Oh, yeah, the DOD research.
Oh, are you talking about the other
I
guy?
remember that. That guy.
Yeah.
The bouncer, not a person.
The thing is he starts with the like question, and then it's like.
How do you know you're not?
I'm knowing you that.
I just know I'm not.
I think that's what she said.
It's not possible.
Okay, good morning, everybody. I think we will get started. Hello to everyone in person and on Zoom. Welcome to the first of this year's Cancer Center seminar series. It is my distinct privilege and honor to be introducing Dr. Carol Lange, who is kicking off this year's seminar series. Carol spent her training years in Colorado. She received her PhD at CU Boulder and then moved on to do her postdoc training at the University of Colorado Health Sciences Center. We actually overlapped there, so I remember her from our training days. She then joined the faculty here at the University of Minnesota and has risen through the ranks, and she currently wears many hats. She is a professor in the Department of Medicine as well as the Department of Pharmacology. She holds the Tickle Family Land Grant Endowed Chair in Breast Cancer Research and is the Associate Director for Basic Sciences in the Masonic Cancer Center. As most of you know, Carol works in the area of breast cancer. She is really internationally known for her work on the progesterone receptor and hormone-dependent breast cancers, which we'll hear about today. Specifically, the integration of PR in both signaling and transcriptional regulation. And more recently, she's been expanding her work into other hormone receptors, which we've also heard about in previous talks, such as glucocorticoid receptor and how it integrates stress signaling in triple-negative breast cancer, and also works in the area of ovarian cancer. And I think that the thing that impresses me the most about Carol's work, and you'll see this through her talk today, is really the way that she integrates these really complex mechanisms involving signaling and transcriptional regulation into these very elegant models that dive really deep mechanistically into defining critical roles for PR and other receptors in key functions of breast cancer like stem cells as well as metastasis. Carol is also highly collaborative. As you'll see, she works both within the institution and outside with many other external collaborators, and it's really fun as a collaborator to work with Carol. Carol, the other thing we have to mention about Carol is that she is quite active in mentoring. She is the PI of the Cellular Targets of Training T30 NCI-funded T32 program. She has a very long list of previous trainees in her lab, many of which have gone on to do great things in cancer research. And I think many of us in this room and on Zoom have also benefited at some point in our careers from Carol's mentoring, including myself. She's always just amazing when you need advice in terms of science or career development or just to commiserate about something. Carol always has great advice and wisdom, and I think many of us have benefited from this along the way. So I'm very excited to introduce a great colleague, scientist, and friend.
Thanks so much. It's great to be here in person, and hello to the virtual audience. So today I thought it'd be fun to focus on progesterone receptors, and I hope to convince you that we should be thinking about targeting these receptors much more seriously to treat women with luminal breast cancer. And I have three sort of take-home messages. Like all steroid receptors, progesterone receptors can integrate multiple signaling pathways. They actually serve as signaling nodes to organize and direct signaling messages to the cell. They're really important for sort of non-proliferative cancer hallmarks. I think we focus way too much on proliferation, primary tumor volume, things like that, Ki-67. And these receptors are regulating other harder to define, harder to quickly measure types of cancer hallmarks. And then finally, I think we can use the phosphorylated receptor as a biomarker of advanced cancer behavior. So when we have a big ugly tumor, we know that we have a hypoxic center, there's high reactive oxygen species, there might be a necrotic core where nutrients are deprived and cytokines are high in the tumor microenvironment. And these harsh conditions are stressful We target these with endocrine therapies, for example, and it may, I wonder if those treatments don't actually support and promote epithelial to mesenchymal transition and actually can contribute to driving dissemination. And so that's a concept I want you to think about as we go through the talk. And that these properties of these cells that undergo EMT include stemness properties and the ability to disseminate and metastasize. And so if we look at the famous hallmarks of cancer wheel and look at steroid receptor actions, steroid receptors are functionally redundant and they drive many hallmarks of cancer. And we kind of think of the estrogen receptor and the androgen receptor as involved in proliferative signaling of either breast or prostate cancer. Progesterone receptors sort of sit in the middle. There's reports that they can either inhibit or promote cancer proliferation, and very much depending on the context and the conditions. They also are immune modulatory. So many of the steroid receptors can modulate immune cells. They're involved in pro or anti-inflammatory processes. They're mediators of metastasis, and there's been recent publications from the Briskin lab, for example, about the role of progesterone receptor in metastasis. They play a role in DNA damage and repair processes. They interact with BRCA molecules. All the steroid receptors have been implicated in pro-survival and also metabolic plasticity. So PR and GR, for example, can regulate metabolism and cancer metabolism. And so, when we think about looking at primary tumors or screening for proliferation, or companies will use tumor volume, they'll use Ki-67, and clinically we're kind of missing all these other 10 hallmarks of cancer when we do that. Okay, and so these cells that are doing these other hallmarks are frequently in G0. They're frequently not proliferating, or they're weakly proliferative. So why should we care about this G0 condition? And as I kind of mentioned, or alluded to, we kind of have a lot of information about primary tumors. We've done a lot of sequencing and omics analyses that are in public data sets. And we've also done a great deal on the other side and looked at metastases more recently. We have a lot of data for distant metastases, but we don't have a lot of data for what happens in between. And so we have this process that happens when cells become less adhesive to the basement membrane. They can either form clusters or go as single cells and undergo this process of EMT. These cells that are becoming more mesenchymal are highly migratory. They're usually non-proliferative mixtures of cells that can include immune cells and fibroblasts, and they have altered adhesion and metabolic properties. And we kind of use these Ki-67 readouts and blocking therapies clinically, and sort of forget about these cells out here. And so we can target the proliferating cells very well. It's very effective. However, I don't think we're targeting the G0 cells very well. And so the data are lacking in that area. So, and as I said before, I wonder sometimes if blocking proliferation with cytostatic therapies actually can promote EMT and dissemination. And I don't have data for that today, but that's kind of one goal of the lab, is to try to delve into that. And then here's a much better picture of this process, where you have all of the steps involved in metastasis. And as I said, we have a lot of data for the primary tumor and the very early invasive tumor. Not much data for this process, and maybe a lot more data for a macroscopic metastasis that's actually transitioned back to the epithelial state or somewhere in between. And so if we're talking about breast cancer now, 1 in 8 women will develop breast cancer. This is a very high rate and expected to increase for many reasons. The major risk factors include age and genetics. We don't really have a lot of control over that. Breast density, hormone exposure, and reproductive history are certainly risk factors. And some of the factors we can control a little better: diet, exercise, lifestyle. There are many more that have been described: life circumstances, stress, And I'm not really going to tell you the GR story today, but it sort of is involved in this, this story. And today I'd much more like to focus on the hormone exposure side of things. And if we look at the types of breast cancer that are have been well defined and are the subtypes that we talk about clinically, the ER positive is the most common, luminal A being most common, which has a lot of ER and a lot of PR. Luminal B tends to have a little bit less ER sometimes and can be PR negative or PR very low. And then triple negative is defined by lacking a detection of either the ER or the PR and does not have an amplified HER2 receptor. And so survival and outcomes are pretty nice for luminal A patients. And then as we get over towards the triple negative, it becomes much more difficult because we don't have these targets that we've defined. And so if we look at these steroid receptors that we can talk about in breast cancer, primarily, sorry, I keep clicking the wrong thing to advance the slide. So the estrogen receptor defines luminal breast cancer, ER alpha. And progesterone receptor is a target gene of ER. So we need the ER and estrogen to get expression of the progesterone receptor gene. So usually the presence of PR is determined as signifies a functional ER. So clinically, when we have ER and PR, we can say the ER is functional. And historically, the PR is simply used as a marker of ER action because it's an estrogen-induced gene. Tumors also contain these other receptors, the AR and the GR, and there's been a lot of data about the increase in AR as tumors become endocrine resistant. And also there are triple negative cancers that contain a lot of AR. And more recently, people have been looking at GR. GR is also found in both luminal and triple negative. And in luminal, GR is very good. In triple negative, it's very bad. So typically, if you have a lot of these receptors in your luminal cancer, you have very good outcome. And as they drop away, the outcome is worse. Okay, so that's a very short and rough introduction. And one thing that we focused on in my lab is the context in which these steroid receptors function. So in breast cancer and all cancers, we have early genetic events that activate signaling molecules. We have oncogenic events, a different signaling environment. And this signaling environment confers an altered hormone responsiveness. There might be altered transcriptional activity and stability of these receptors because they're heavily modified by protein kinases, for example. And that would lead to a different promoter selection, a totally different transcriptome, different biological outcomes, altered cell behaviors, and even altered cytoplasmic properties, which is one area that my lab has focused on over the years. And so why should we care about progesterone receptors? So I just jotted down a few things off the top of my head. PRs are major effectors downstream of the estrogen receptor. You can think of them as a major effector of the estrogen-driven pathway. Again, the PGR gene is estrogen-induced. Progesterone receptors are the primary drivers of proliferation in the normal mammary gland. Estrogen is permissive for that action because it increases the PR. And it's really the PR that's driving proliferation in the normal mammary gland. Progesterone and progesterone receptors are required for mammary gland stem cell expansion. And that work has also spread into the cancer arena. So progesterone, but not estrogen, drives stem cell expansion. And the target genes of PR include a lot of secreted factors, WNT, rank ligands, cytokines such as IL-6, ligands for receptors such as EGFR. And so a small number of cells with PR can do a lot of damage because they're secreting all of these factors. And historically, they can signify an activated ER. And the reason I write this here is because we already have the infrastructure to look at PR. This is already clinically evaluated with clinically used monoclonal antibodies. So this is already something we already do in the clinic. And ligands exist. So these are ligand activated transcription factors. And so there are lots of ligands out there. So they're highly druggable. We have things like antiprogestins such as mifepristone or onapristone. And these are coming under fire because of the anti-abortion fight in this country. And so I think companies have been really hesitant to develop these reagents because of that. And then clinical trials have been very encouraging. However, specificity remains a challenge. So we don't really have ligands that can target the PR specifically. They also target the GR and the MR because their ligand binding domains are very similar. And so we lack a collection of ligands that are highly specific. And so we can see some toxicity. Most of the studies with antiprogestins are the danger is liver toxicity, probably because it's engaging the glucocorticoid receptor and causing some toxicity. So I think we have a long way to go on getting ligands. So one thing in my lab that we'd like to do is maybe we can't target the progesterone receptor right now because GR and MR are much more ubiquitous than PR. But we can identify novel molecules that are in the complex and required for the action of PR, and we can target those. So that's one of the reasons that we're doing what we're doing. And then just some background on PR. So all steroid receptors are found in both the nucleus and the cytoplasm, and they rapidly and dynamically shuttle between those two compartments. And this shuttling allows this beautiful crosstalk with signaling molecules to occur. And that's been well defined almost 30 years ago, where both ER and PR are found in a complex with Src kinase, and then steroid interaction, either progesterone or estrogen, can activate MAP kinases very rapidly, and that provides a mechanism that can happen in minutes and regulate transcription through the MAP kinase pathway. And then the other reason this happens, I think, is because you want to specifically phosphorylate these receptors to direct their transcription. And so this is very much understudied, and now we know that ER and PR are also in transcriptional complexes together. So I think people are starting to pay much more attention to this rapid signaling function of steroid receptors. And this is one part of the pathway that my lab has spent a lot of time defining. And so we think that actually these steroid receptors are recruiting kinases for the express purpose that they're going to be directing specific phosphorylation events on both ER and PR, and that those phosphorylation events are going to translate to promoter selection. So now the heavily modified receptors are going to target specific genes based on their phosphorylation events. And they cooperate with other factors such as AP1 and SP1 when they're phosphorylated. So it provides a really beautiful sensing device for what other pathways might be activated in the cell, and then drives the receptors to different places in the genome. And more recently, we've defined this IRS1 molecule as being in the complex, and I'll touch on some of that data today. So, okay, so we became very interested in these phosphorylation sites on the progesterone receptor, and we've published many papers on these 4 sites. There's 14 sites total, but today I'll focus on this serine 294 site. So we have 2 isoforms of the progesterone receptor, PRA and PRB. PRB is the long form, PRA being the short form, which is created by an internal translational start site here. Most normal tissues have a ratio of 1:1. Luminal A tumors are known to be PRA rich, whereas luminal B tumors tend to be PRB rich, even though there's much less PRB or PR. Claudia Linari has defined that they are PRB rich. And we define that these phosphoreceptors tend to be very hyperactivated in low dose progesterone. So I'll just be focusing on that site. And so we decided to make an antibody to that site and look at tumors, and we did that with Kaylee. And we found that a majority of ER positive tumors had this phosphorylated residue on PR. Here's an example of that TMA, where over half at least had over 20% of this phospho-PR, and you can see the total in the phospho. But we found really no correlation between PR staining with this monoclonal antibody and our phosphostaining with our polyclonal. And so we suspect that when the receptor is heavily modified, the monoclonal site could be occluded. And so we did things early on like this, where we'd take cells and, for example, treated them with estrogen. And so we treated PR null cells and cells that had PR with estrogen. And we found these interesting genes that were either up or down regulated by estrogen, but required the PR for their estrogen signal. So we're getting back to this ER and PR complex idea. So we defined this 40 gene signature that basically was estrogen induced, but required the presence of PR, much like a scaffolding molecule. And we validated some of these genes. This is the work of Truong, which is unpublished. But you can see that these genes are in the literature. Many of them inhibit cell proliferation, but drive stemness properties. So we thought that was quite interesting. And these were PRB regulated genes that needed, were estrogen driven, but needed PRB. And if we look at all 40 genes in the public data, we can see that if patients express these 40 genes that require PR for their estrogen induction, they do very poorly on endocrine therapy. So this is like a bad gene signature. And we think that we validated some of these, and we think that we have an ER and PR complex coming to an ERE site on these genes and requiring PR for that specificity. We've now narrowed this down to, hold on, not working. Oh, it's just taking a while to load this slide. Okay. We've narrowed this down to a 15 gene set signature. These are 15 phospho-PR target genes that confer endocrine resistance. And this is one example from some PDX models where we collaborated with Carol Sartorius at Denver. And what's interesting about this, and the reason I'm showing it to you, if you look at total PR in these two different PDX using our monoclonal antibody, it is really not that impressive. You really would say that there's very little total PR there. However, if you look at the phospho-PR, these are two different heavily treated patients. One has a mutation in the ER gene, and the other one has an amplified ER. So they have a lot of ER. But now if you look at the phospho signature, it is much more impressive. So again, we think the modified receptor is just not being seen very well by the monoclonal used clinically. And then if we look at these metastases in these models, they had a lot of phospho-PR. Not every cell had it. It was quite focal. And then Carol's lab did single cell RNA-seq on these tumors, on these metastases. And remarkably, they found that PR and PR target genes were quite elevated in the single cell data, and genes that are known to be downregulated by progesterone were downregulated. This would not be visible by bulk sequencing. She was only able to see this by single cell. And then we've played around with this antibody. I just want to show you some of Laura Morrow's work in fallopian tube and these serous tubal intraepithelial carcinomas, or stick lesions that appear in the fallopian tube. Just to kind of show you this antibody a little more detail, is again, we can see total and phospho-PR in these stick lesions that are defined by P53 mutations. And if it's taking forever to load these images, sorry guys. Oh, that looks weird. I don't know why that's there, sorry guys. It did not work. Oh, that really didn't work. I just wanted to show you that we really see no correlation between phospho and total, and especially during progression. So this must be a Mac to PC thing. But during the progression of these stick lesions, we lose the signal for the monoclonal total. We gain the signal for the phospho-PR. They're highly focal and nuclear, meaning we think they're activated. And so this is just an example of validation of that antibody in a real clinical setting. And so here's what we think what happens is ER is needed to express PR. We get the PR. PR can actually inhibit the activity of ER, and it does that by sumolation. And this is coming out of the literature. However, these protein kinases that are highly activated in tumors modify the PR. It gets phosphorylated. It lets go of that sumo, and you get a totally different program that's driving dissemination, and the antibody no longer can see it. And so that's one of our hypotheses that we're working on, and summarizes a great deal of work to get to this point. And so now I'll just show you some evidence for some of that. So again, Tu, who was in my lab, looked at the ER-PR complex, and she learned that the ER-PR complex can only form when you have that phosphorylation site intact. And so when you mutate that serine to an alanine, you no longer have a really tight interaction in these IPs. Then she also looked at proximity ligand assays. We published this, but if you treat with estrogen, you can see the formation of this PR-IRS1 complex in these foci here. So treating with estrogen, we can recruit IRS1, which also showed up in our single cell seq. And when we mutate that MAP kinase site on PR, we completely lose the interaction. And so now we have this ER-PR complex, which is estrogen driven and can recruit IRS1 into the nucleus. And then we have this model where we have growth factors causing the desumulation of PR. Normally this sumulation is a repressive mark. And then when kinases come in, they release that sumo and we get a different complex, sort of a release of inhibition type of model on these genes. And now we're looking at this IRS1 role in this recently published work, and I'll just show you a couple examples of that. So we decided to make the sumo mutant PR, where we made a phosphomimic by getting rid of the sumo site. And you can see that data here. When we mutate that lysine, which is the sumo site, now we have a de-repressed receptor. It becomes tamoxifen resistant. So here's two different cell lines where we created the K388R receptor, which is desumulated, mimicking a phosphorylated receptor in our hands. And you see that it's TAM resistant in two cell lines. These cell lines, interestingly enough, became very sensitive to insulin. So you can treat them with insulin and show that the phosphomimic or sumo mutant receptors, cells with those receptors, are much more prone to growing in insulin, much more sensitive. And this is kind of a hallmark of endocrine resistance, that endocrine resistant models tend to have increased IRS1, loss of the IGFR receptor, and the fetal insulin receptor signaling. And Dr. Yi has done a lot in this area. And we saw increased stem cell markers. So the cells with the sumo mutant activated receptor had more aldoflor and CD44 high, CD24 low, which are stem cell markers, and they grew out in mammospheres. And then we could inhibit this process by inhibitors of the IRS1 molecule and also the ER alpha receptor. We then showed that even the endocrine resistant cells that had endogenous progesterone receptors were growing much more readily as mammospheres, and this is like a stem cell, poor man's stem cell, or poor woman's stem cell assay in vitro, where you grow out primaries into secondaries, and then that can be an indirect measure of stemness properties. And they have a little bit of PR, a very little bit, and you can see that there, PRB and PRA. However, as we grow out in these 3D models, the PR is inducible. So it's being induced as they grow out. And you can say, well, is that enough? We can see it very readily in flow cytometry. This is the phospho-PR in flow cytometry, and then PR target genes are increased. Those are all PR target genes that we've identified in our arrays. And then we can see the proteins for those. I showed you messenger RNA. You can see PR target genes at the level of the protein. When we knock down PR, we can inhibit the tumor sphere growth, and we can really nicely inhibit that by inhibiting the IRS1 molecule, as well as progesterone receptors with antagonists. And the T47D model tends to be pretty resistant to onapristone. So coming in with mifepristone, you see much better inhibition. So we can target these pathways and inhibit this stem cell biology pretty well. We've even gone farther to endocrine resistant models. So these are from Tony Diosoro at the Mayo Clinic, where he made these in mice, where he injected cells into mice and then treated the mice with letrozole or fulvestrant, either separately or together. And these are his double resistant to both letrozole and fulvestrant. So an anti-estrogen and a aromatase inhibitor. And he cannot detect PR in these cells by Western, but we can detect it by flow. So we can see it, much more sensitive assay to use flow cytometry with our specific antibodies. And so we can see the parental, the letrozole only, and the double resistant all have phosphoreceptors. So we made a knockout of PR and gave these to Tony. And again, the images take forever to load. Come on. There we go. Tony injected 50,000 cells into these mice, and you can knock out PR here, the PR target gene STAT3, or the PR target gene KLF4, which is also a stem cell marker. And you can see that these tumors are much smaller in these mice. And so we now have this ER-PR complex, we believe, that's regulating stemness genes, and we defined a complex with PR with aurora kinase and STAT3 that's actually driving the expression of those same genes. You can reduce that to 5,000. Let's see. Come on. It's really slow. Maybe it's good. It's slowing me down a little. It says it's loading. This does not happen on my computer, so sorry, guys. Huh, doesn't like that. All right. Well, we have very similar data when we go down to 5,000 cells, and I was going to show you the quantitation of that. Do we want to restart? Seems to only do that on when I have images.
Yep.
I'm way ahead of time, so I'm just talking too fast.
All right, let's try it again.
Thank you.
Come on.
I tried going backwards too, and it didn't like that either. It's like when you're watching a Netflix show and it
Is that the one?
starts to do that. No, go back a little bit. That's good. And then is there one? And then can you go forward now?
That's the one you want?
Yep.
All right, let's try it again. And let me put this back on Zoom. Come on.
Those files might be really big. Is that the problem?
I think that kind of is it. Let me, that's what I want. That's what I want.
That looks good.
One more second. Let me put it back on Zoom.
No, it doesn't like you.
It doesn't.
Oh, there it
There it
is.
is.
I lost it again. There it is.
What we
Go
wanna
slow
do is-
there. Right there.
Now we want to go to here and go there.
Now, are we on Zoom?
Yes.
Thank you. So, that was a little breather. Sorry, guys. All right, so let's just see what I was saying. So this is just a look at those tumors and the quantitation. And then very similar data were observed at a much lower injection level of 5,000 cells, again with PR knockdown. So if you knock down PR in a model where you cannot see PR by Western, this is what you get. And Tony has gone on with my lab, Carolyn Deep in my lab, to look at some of these genes. And so this is chromatin immunoprecipitation assays on the KLF4 promoter, where we can see recruitment of aurora kinase in blue with progestin and blocking with the aurora kinase inhibitor. We see phospho-STAT3 coming in to the same place and well blocked by alisertinib, the aurora kinase inhibitor, as well as the phosphorylated PR, which is coming into this site as well. So we think now in this model, we have STAT3, which is a PR target gene, aurora kinase A, which is a PR target gene, and PR, phospho-PR, assembling on PREs upstream of the KLF4 promoter. And KLF4 is a marker of sort of STEMI-like cells. It's been defined by Carol Sartorius, and so it's really been a nice gene to look at. And then, of course, the mRNA is also regulated. And then Tony used the IncuCyte machine to look at these cells, where if you treat with both onapristone, which inhibits PR, and the aurora kinase inhibitor, you see in purple that you inhibit the growth of these cells in the IncuCyte assay. And then most impressively, I think, is this: we are now killing these cells. They're no longer cytostatic. And so this is a pretty low dose of onapristone, and onapristone alone wasn't that impressive. But when you add them together with alisertinib, you can see a really nice apoptosis signal coming from these cells. And so Tony at the Mayo Clinic is designing clinical trials based on this idea. They're already doing alisertinib alone and looking for ways to combine alisertinib with other agents. And I think antiprogestins are attractive because low-dose antiprogestins are pretty safe and have been around a long time. And we may not be able to treat people with high-dose mifepristone or onapristone, but low-dose might be a really good combination therapy. And then looking at a couple other endocrine-resistant models, we kind of switched around to different endocrine-resistant models to see if this was a pervasive theme or just kind of a one-off. We also have a number of these estrogen receptor mutant models. So the patient-derived xenograft I showed you was one of the estrogen receptor mutant models. This is a cell line model that has the Y537S, which is a major mutation in the estrogen receptor that arises during aromatase inhibitor therapy. And again, data from 2 shows that these cells make soft Auger colonies with estrogen, but when you knock out the PR, it goes very low in both the wild type and the mutant receptor. And when she injected these into the MIND model, which is the mammary intraductal model, where you inject the tumor cells into the duct of the mammary gland, she could see that when she collected the blood of these mice, that there wasn't a really big effect in the wild type on getting rid of PR. But when she had the mutants and knocked down PR, there were a lot fewer circulating tumor cells. So she collects the blood and grows out the circulating tumor cells in soft Auger, and that's how the experiment is done. And you can see a heavy dependence on the PR when the estrogen receptor is constitutively active and resistant to endocrine therapy. And then finally, another separate model from Julie Ostrander's lab. Julie has been working on this PELP molecule for many years, and together we had defined that the PELP molecule is recruiting both the estrogen receptor and the progesterone receptor. And this PELP molecule is really interesting. It's a nuclear protein that becomes more cytoplasmic in women with high risk of getting breast cancer or in breast tumors. And Julie made models of cytoplasmic PELP to define how that works. And she identified SRC3 in this complex, which is a co-activator of ER. And this summarizes a great deal of work also. And we think that this PELP, when it's cytoplasmic, sort of pulls out these steroid receptors into the cytoplasm, and they talk to signaling molecules much more, and they become more phosphorylated. It's drawing in the coactivator SRC3 together with Tutruong. They found that it's attracting this phosphofructokinase molecule, KFB4. So this PELP is kind of forming a signaling node. We also define this for GR in triple negative. So it's a big player in steroid receptor action in all kinds of breast cancers. And when she got this PELP out into the cytoplasm, she found that it mediated therapy resistance. And the pathways were metabolism and stemness pathways. And they just published this paper where it represents a multi-investigator R01 between Julie and I, and Tutruong's the first author of this paper. And one of the main things she found is that it's really regulating metabolism. And so when you push PELP out into the cytoplasm and it brings ER and PR along with it, you're getting a very altered metabolism. And really exciting data, I think, is in the MIND model. You can see these primary tumors when you inject cells into the duct. And you really don't see a huge effect on the size of these primary tumors. So, and then she knocked out this PFK, and it's very modest, I think, in the primary tumor. However, the circulating tumor cell data is really impressive. Look how big these colonies are, these clusters are, and when you knock out the PR target gene, which is this PFKFB4, you see this reduction and a dramatic reduction in the circulating tumor cells. So again, kind of getting back to the idea that we're talking about these other hallmarks that may not be proliferation. So a weak effect on the primary tumor with a really large effect on dissemination for the phospho-PR in several different models that I showed you now. I think 4 different models I showed you of endocrine resistance where this is a theme, emerging theme. Okay, so then just to kind of wrap up, we talked about these big ugly tumors and the fact that we think we understand these at least a little better than these cells that are out in the circulation. Targeting the ER is the standard of care and it works very, very well. However, we do have a very high recurrence rate, especially after 5 years, and especially for node positive women. As much as 40% of node positive women could recur, and they recur late. And so we have these cells that get out early into this dissemination and are not targeted by this therapy because it's primarily cytostatic. And then we have this small population of cells, I think, that get out that have phospho-PR. And we're looking now at human blood to try to see if we can see them in collaboration with Dipali, Sachdev. And these cells, we think PR, phospho-PR is driving the stemness properties and metastatic behavior. And now in several models, we're looking into this at the biochemical level and at the genes themselves. And we've identified numerous proteins that we could target. We could target PELP, SRC3, IRS1, phosphofructokinases. All of these that are in the complex could be targeted. And so finally, in summary, I hope I showed you a little snippet of how these phospho-PRs are integrated with the estrogen receptor signal. And another push in the lab is to look more carefully at the insulin receptor signaling integration. Why is IRS1 in the nucleus? What is it doing there? It's not really defined as a nuclear protein. And how is it phosphorylated itself? And what is it recruiting? That's a whole nother project that one of my students is working on. These are driving these non-proliferative pathways. And I think we're really good at killing cells that are cycling, but we're not so good at killing these so-called quiescent, senescent, or dormant cells. And I would say that the word dormancy is not really a good term. These are highly metabolically active, transcriptionally active cells. They're just not cycling. And we need to kill them also. And that we could use the signal or the gene signature of phospho-PRs or the presence of its target genes as biomarkers of this potential that these cells have that's lurking. And then finally, there's a whole army of people, not only in my lab, but also these collaborators that made this work possible. And my sort of PR dream team has been not only Julie Ostrander, who's a great collaborator, but Tu Truong, who just got her own lab last January, who did a lot of the work I showed you. Amy Dwyer went on to take a junior faculty position in Adelaide, and Carlos is an MSTP student who graduated and is now off doing doctorly things out there, right? So, and I think that's it. And I'm sorry I went so fast. I hope it made some sense today. It's been a while since I did this publicly, and it's great. It's great to be here. So thank you very much. Questions?
I have microphones.
Hi, May.
Thank you. Carol, that was fantastic. It was very provocative, so you are officially a provocateur. I have three quick questions. Two are biology, which I love the biology you showed us, and one is technical. So the first one is, you've done a lot of work looking at kinases. Have you looked at the opposite end, phosphatases?
So
let me lay out the three questions,
okay.
And you can.
Phosphatases?
Yeah, phosphatases.
I'm not going to remember them all.
Yeah.
Okay.
The second one is technical. So we early in my career, I spent a lot of time looking at cell cycle, and particularly the G0 to G1 transition and G1 to S. And we looked at RB, and we saw a very similar phenomenon that you described with antibodies, where antibodies against total RB, particularly monoclonals, were kind of crappy at detecting protein. And then we looked at phosphoprotein antibodies, and they were amazing. They were very specific, and they worked on Western, and they worked on IHC, and then the bunnies died, and the antibodies disappeared, and it was the end, right? And so I wonder how much of that is mouse immunology, rabbit immunology, antibody selection, affinity, multiple epitopes recognized by, not multiple epitopes, but multiple antibodies recognizing the same epitope in the polyclonal antiserum. So, you know, maybe I'll leave it at that, because I myself forgot what my third question was.
Yeah, so, you know, this took a long time to figure this out, because I would say the first 5 or 6 batches did not work in anything but a Western. And then we got this beautiful batch that then suddenly worked in IHC and IP and chip assay. And so I don't know, some magic happened with the company we're using now. The last couple batches have been great. And everyone says, why didn't you make a monoclonal? Why didn't you make a monoclonal? Well, okay, I don't have $50,000 lying around. And I'm really worried that a monoclonal is not gonna be as sensitive at all. The purified, so we get the polyclonal, and we have the company purified against the phosphopeptide, and that's working pretty well. I know that isn't really translatable to clinical settings. So yeah, it's a big problem. And so Elaine Allerid gave a beautiful talk at Endo, and she had a table of all the steroid receptors, and they all have a MAP kinase site. So for ER, it's serine 118. For PR, it's this serine 294 that I talked about. And it's a propolysomerase site. And so what she thinks happens is it gets phosphorylated, and then a propolysomerase comes along and twists it, and twists the whole molecule. So it's a dramatic structural change, very big structural change. And it wipes out the monoclonal for ER also, she says. And so she had PR in the list, and she had that site. And then she has a couple of review articles about this, but you know, she said, hello, clinicians, the ER is actually, the monoclonal clinically used ER is being wiped out by this propolysomerase change. And those propolysamerases are highly expressed in breast cancer. So they are making the steroid receptors invisible. And then phosphatases are very complex. We have worked in phosphatases. We did have 2 or 3 papers with MAP kinase phosphatase 3. And again, we found that it binds to PR. It only binds to PRB and not PRA. And it recruits CK2, another kinase. It doesn't do anything to PR, it doesn't dephosphorylate PR. It acts as a scaffold for casein kinase 2, which phosphorylates a unique site on PRB. And if you mutate that site, PRB becomes PRA. So that's the serine 81, where you can totally make PRB into PRA by getting rid of that site. You know, so again, it's another complex biochemical story. how these receptors are incredibly flexible, malleable. I think it's because they're so ancient and they're required for reproduction. And so you got to reproduce. And so the trade-off for that reproduction, that success, is breast cancer and prostate cancer. It's so important to reproduce, right? And so they're ancient molecules that can adapt to almost any situation and function, which is fascinating, right? It's just fascinating. But so phosphatases, yeah, I haven't really, a lot of them are gone in cancers, less.
If you remember back to when I were young and still had memory, we did a little bit of work in the, yeah, speak for myself, right? Well, I've known you for at least a couple of years, yeah. In the cat model that we played with way back in the late 80s, there was a PR mediated, I don't know how many steps in between, reduction in phosphorylation of HER2 and EGFR that seem to be sensitive to phosphatase inhibitors, right? So this is ancient paper, ancient technology, and I've always had the curiosity about, kinases are easy to look at, phosphatases are hard, and if we're just missing that other half of the story.
Yeah. So when I first met Jaime, I think we biked over the pass from Copper Mountain to the next town, and we took my poor thesis advisor with us. And I think he couldn't walk for the rest of the whole meeting. He was gone for like 2 days. Doug has a question, or I'm sorry, Scott has a question.
Yeah, I'm curious about the phospho-PR gene signature and those Kaplan-Meier survival curves that you were showing. Did the patients that you used as input for that Kaplan-Meier analysis include all subtypes of breast cancer, or were these ER positive patients specifically?
In those early ones, they were ER positive.
Okay.
In the more recent ones with GR, it's all comers, but the PR ones we do mostly luminal.
So is there any overlap in those gene lists of phospho-PR targets with genes that define other breast cancer subtypes?
So the biggest
Specifically about
thing we always have seen, which we never understood, I mean, we were one of the first people to show the ER-PR complex in tumors. But so we'd always get these gene signatures that were not defined as PR, they were defined as estrogen. That's why we started using estrogen, because we'd be like, wow, these are all estrogen target genes in our P. So a lot of those phospho-PR target genes don't really respond very well to progesterone or anti-progesterone. They're very sensitive to estrogen. So it's like a scaffolding action of the phospho-PR. And so a lot of those genes aren't in any gene list for progesterone, and so they don't come up. And so that's why we kind of switched to using estrogen as our ligand, and then having no PR knockdown or something, or the phosphomutant site. And so, it's just totally unexpected.
Well, it's very clear that you're picking out a bad subset of these ER positive patients.
Yeah, and those are all estrogen signatures, really, in the literature. They're estrogen signatures.
What's the correlation between total phospho antibody detection and mRNA? And I'm asking, most of the tools that clinicians use are mRNA based now, Oncotype, Mammoprint, PAM50. PR is measured in all of those by mRNA.
Yeah, it's low. Yeah.
So it's low while phospho is high?
Yeah, so I think that, so we have all these different models, and we really wouldn't have seen that if we hadn't done single cell. So Carol Sartorius and I think that we have got, as the tumors progress on endocrine therapy, there's probably a very small, in a heterogeneous tumor, there's a small population of cells that still have PR. And it's those cells that are PR positive, that are rare, that are able to disseminate and grow out later. So it's probably going to be, so we don't, like I showed you that Western, it's very hard to detect on a Western, the PR. And so there isn't very much there, but all of the factors are secreted factors. And so, it becomes really difficult now because you can't see PR in those genes in the mRNA level. It's low. Yeah. And it's inducible in 3D. So as you grow out the stem cell compartment, you see more and more of it.
Yeah, so they do probably score in the luminal B.
That luminal B.
Is luminal B.
Yeah, so Claudia Linari has done the most of this, and she's found luminal B has PRB rich. And we tried to work with her to get those, to look at phospho-PR in those, and we just didn't get very many, and the quality of the lysates wasn't very good. We've also worked with Helgar Rui, and he did see the phosphostat-3 was very high in the phospho-PR subpopulation. In his, he had a technology for looking at that. So that, I mean, that's still very hard to do because it's going to take single cell technologies to do all that. I mean, because it's hard. I mean, that's what, it's hard to argue with people when they say, well, it's not showing up. The mRNA doesn't show up. It's like, it shouldn't show up because it's going to be it's like 1 in 1,000 cells probably.
Hi, Carol, Emmeline Alejandro in IBP. I just came back from Italy for a meeting for developmental origins of health disease, and there were several talks there that suggest that there's a fetal origins of breast cancer. So there's also strong correlation between obesity and cancer. So I just been thinking, and there's also now data suggesting that girls are just gaining weight at a much earlier age, obesity, and this is linked to hyperinsulinemia and inflammation. So I was just wondering what your thoughts are in terms of, you think that insulin is driving part of the susceptibility perhaps, or even priming. You did mention that there's that interaction between the PR and the insulin signaling pathway, and I was just wondering if people have looked at predisposition in models that would show.
I would say the most sophisticated people are Gail Prenz at University of Illinois and Cheryl Watson, she keeps moving around in Texas, but anyway, she has done these, so they've done these mouse models, and when female pregnant mice are exposed to environmental estrogens, so endocrine disrupting chemicals, which are ubiquitous and increasing and everywhere, plastics, all that stuff. Those offspring have a much higher mammary cancer and prostate cancer in the different models that she's used. And it is methylation, not mutation. And the methylated genome is past 9 or 10 generations without change. And the prediction is from, Catherine Briskin's done some of this, the predictions are that this 1 in 8 of women is actually going to rapidly go to 1 in 5 because of the methylation signature that's conferred by environmental estrogens. Environmental progesterones have also been defined by Joanna Burdett. And I'm sure there's androgens as well. So it's a big problem, huge. And what it's doing is changing the number of stem cells. So the mammary onlaga, so mammary glands in mice aren't formed until puberty, or till pregnancy in mice, and humans and rats at puberty. So there are more stem cells present in the offspring of these rats and mice in the prostate and breast when they've been exposed, the dams have been exposed to these chemicals. And so the idea is that there's more stem cells, there's more cells that can give rise to cancer.
Sorry, I'm not an expert in mammary gland, but is that the same in human? You mentioned that
So
is the case in
humans
the
are
rat.
more like rats, where the
Okay.
mammary gland develops after birth during puberty. So, you're trying like, well, okay, the exposure happens in utero, but the number of stem cells you're born with increases, even though the mammary gland doesn't develop until puberty. So, and then all of those cytokines in that pro-inflammatory environment in obesity all activate these receptors, all of them. They're all targeted by the P38 MAP kinase. Those are MAP kinase sites, but any MAP kinase can hit that site. So it doesn't have to be ERK1 and 2. It can be this P38, which is cytokine. So all of those hormones can. What happens is the phosphoreceptor can respond to very little hormone. Still needs hormone, but we showed in vitro that normally progesterone receptors need 10 to the minus 8th molar progesterone. When we have MAP kinase there, phosphorylation, now it goes to 10 to the minus 14th on a reporter gene. which is not very sensitive. So you just need a drop of hormone when it's phosphorylated. It's incredibly sensitive, hypersensitive. And so I do think the role of the pro-inflammatory tumor microenvironment and, you know, like obesity would be to very much lower the amount of hormone needed to misregulate these receptors. And so they're on all the time instead of being regulated.
Okay, we'll take the last question for today at the back here.
Hi, thank you for the nice talk. My question is, although PR is an ER target gene, are there any alterations to estrogen receptor signaling by the virtue of maybe PR antagonism? For instance, for patients that take mifepristone, do you expect that ER signaling is altered in any way such that ER positive breast cancers could benefit from those at all?
Yeah, that's a really good question. I don't think anyone's tried that. I mean, because most people who take mifepristone, the clinical implications for that are, you know, for termination of a pregnancy, and then sometimes it's given for a rare type of brain tumor that has a lot of PR in it, called a meningioma. So there aren't clinical data for, there's not really a clinical indication for antiprogestins alone, or for progestins alone. They're given with estrogen as part of hormone replacement therapy. But I would predict yes, because of the interaction. Now we have to think of ER and PR as a complex. Because of the interaction, modifying one will modify the other every time, I think. So definitely, but there just aren't clinical uses for these other than in hormone replacement. And there aren't that many out there that are being used. There's some clinical trials I'm aware of. And the one clinical trial I was involved with was terminated because of liver toxicity, probably because of the GR. And then Semacon, so people with BRCA carriers have elevated estrogen and progesterone, all body. Their ovaries make a lot more. And so Semacon in Chicago has been doing prevention as well as clinical trials with antiprogestin tilapristone in BRCA patients that she sees. She's an OBGYN there. And I don't know how those are going.
That is fantastic. Let's go ahead and say thank you one more time to Carol for a great talk.
There needed a lot more coffee.